these in various organisms. The molecule consists of a nucleotide the RNA and DNA polymers are made up of long chains of nucleotides. And a nucleotide consists of a sugar molecule, which is either ribose in the case of ribonucleic acid or deoxyribose in the case of DNA, attached to a phosphate group and a nitrogen containing base. And in RNA, those bases are shown here: cytosine, guanine, adenine, and uracil. The difference here is that in DNA, uracil is replaced with thymine. So the COVID-19 virus, like all viruses, cannot replicate on its own. What it needs is to be inside a cell where it uses its own genetic code or RNA to take control or reprogram the cells, thus making them sort of virus-making machines. So the COVID virus would attach to a cell through an ACE2 receptor, inject itself into the cell or its RNA into the cell, replicate within the cell to produce additional viruses. What happens is that these cells end up dying, and then the viruses are released and then can go on to infect more cells. We know it's a new virus, or one that we have just recently become aware of. So our bodies are just simply not equipped to handle, or our immune system is not equipped to handle this virus very well. So what about the role of chemists? Like I said, we are, I think, mostly chemists here. So I wanted to spend a little bit of time on the role that we can play in this fight against COVID. And in fact, we play very important roles in fighting many diseases, including COVID-19, and I have some of the roles listed here. It starts from the analysis of the genetic material or genome, which is then used to develop testing and detection protocols, to the development of surfactants and soaps for cleaning, disinfectants for sanitizing. Marvadine would like this area, material development for the prevention of the spread of the disease. And I don't know if anybody else saw it, but I read an ACS article just yesterday, would you believe, that discussed how scientists around the world are developing antiviral masks that can both trap and destroy viruses. That's a very interesting area of research, I think. We're also involved in the development of drugs and vaccines, and there are many other areas of research and development that chemists are involved in. The areas of expertise that we have here in the department can be put to good use in this fight, and not just in the department, but in the faculty. Of course, we have persons in natural products research for drug development, occupational and environmental health research, material science, as I mentioned, food research, and of course, my area, environmental studies. So it leads me then to the focus of my talk today, which will look at the detection of the virus in the environment and how this may help us predict outbreaks and track the spread of the disease. So in particular, I will share with you some research that is going on across the world, looking at the testing and prevalence of COVID-19 in water and wastewater. So some questions that have been asked, so I'm going to try to answer them for you. They're listed here on the slide. Some of the questions I've been asked include: how do we test for SARS-CoV-2 in water? What is wastewater-based epidemiology, otherwise called WBE? Has the virus been detected in various types of water bodies, including rivers, streams, groundwater, and wastewater and sewage? Is water a route of transmission? Are our drinking waters and surface waters safe? What can we do to protect ourselves and the rest of the population? And can we use wastewater-based epidemiology in this fight against COVID? So let me start with the first question, and let's look at how we test for SARS-CoV-2. So there are three main tests used to detect COVID-19. The first one that I'm mentioning here is one that I think everyone is familiar with, even if you don't know the nitty-gritty details. It's the viral test, the PCR or RT-qPCR test that is used very often. This viral test detects the presence of whole or fragments of the virus, and this test is carried out on nasal, throat, or saliva samples, so swabs that are taken of these, as well as water and other environmental samples. Because this test can detect both whole or fragments of the virus, then when a test comes back positive, it means the virus may be alive and viable, or it may not be alive or viable. This PCR test is the gold standard for detection of most viruses, including COVID-19 or SARS-CoV-2. Another test you've probably been hearing a lot about is a rapid antigen test. And this is also a test done on nasopharyngeal or nasal swab samples. In this case, the test detects viral antigens or proteins. And basically, if the virus antigen is present in high enough concentrations, then it will bind to a paper strip that is enclosed in a plastic casing and generate a signal that you can see, typically within 30 minutes. This only happens, by the way, if the virus is actively replicating. So it is best used during early stages of infection when virus loads are high. It's a relatively inexpensive test though, so it can be useful. The problem is that it is most effective within 5 to 7 days of the onset of symptoms. Otherwise, a virus load may be below the detection limit and therefore lead to false negatives. It is less likely though to give you a false positive, but that can happen if other coronaviruses are present. So this test is useful, but again, as I mentioned, it's really most useful when viral loads are high, and it is fraught with false negatives, a lot of false negatives that can come out from this test. The third test, which you've probably also heard about, is the antibody test, and this is a test carried out on blood samples, and it can be used to tell if there has been a past infection. The thing with this test as well is that it could also be negative even when infection is current, if your body has not yet developed antibodies that can be detected in the blood. It may also be positive for other coronaviruses. So, if we compare the three tests, the PCR test is the most sensitive and therefore is the gold standard. The rapid antigen test can be useful, but only in certain circumstances, and the antibody test is useful after to really show if somebody has had the infection. As a result, the WHO does not currently recommend the use of the antigen or antibody detecting rapid diagnostic tests for patient care, but rather encourages further studies of its potential use in this area, and certainly encourages the use in surveillance and research. All right, so what is this gold standard RT-qPCR test? In this case, we're looking at the coronavirus, or nCoV, so it's a novel coronavirus 2019 nCoV real-time reverse transcription or transcriptase PCR diagnostic panel. And the Q that is mentioned in the RT-qPCR, RT means reverse transcription, real time is, we use the word, or the Q is used, or quantitative or real time PCR. It is the most widely used method, and it is used, as I said, to detect SARS virus in upper and lower respiratory specimens and environmental samples. To detect the COVID virus, we rely on specifically detecting a sequence of RNA that is unique to that particular virus, and this works for all viruses. Using RT-PCR, the RNA virus is converted to complementary DNA using or via the process called reverse transcription. And this is done because only DNA can be copied or amplified in a process called PCR, or polymerase chain reaction. So analysts then amplify a specific part of this transcribed viral DNA hundreds of thousands of times. And what that does is, even if you have a small amount of virus in the sample, it means it can be accurately detected because of this amplification. The method uses the same RT-qPCR testing instrument that is commonly used for other viruses, such as influenza, Ebola, Zika. The difference here is the primer and probes that are needed, primers and probes required for the COVID-19 virus. So in this method, the sample is treated with several chemical solutions, including buffers and other chemicals, to extract the RNA present in the sample, convert it to complementary DNA, and then amplify and detect it. And these primers and probes have been developed specifically for the COVID-19 virus. And a list of these are actually available on the CDC website for those of you who are curious. That's not my area of research, I'll be very honest with you. This is where I rely on the biochemists. Normally though, the demand for these enzymes would be steady. All these chemicals, enzymes, buffers, etc., would be steady and relatively low. But because we're faced with a pandemic, we're having serious issues keeping up, because manufacturers then have had to scale up the massive quantities that are required for this COVID-19 testing. And that takes time. In addition, like I said, the new primers and probes are required, and we have to ensure that the tests are accurate, right? That they're not giving us false positives or false negatives. So tackling all of these issues has been very difficult and has certainly contributed to some of the delays we have seen in testing so far. So as I mentioned, this is the test that is used not only in nasal, throat, saliva samples, but also in environmental samples such as water. So what is wastewater-based epidemiology? And that is the focus of my talk. WBE, or environmental surveillance, has been attracting global attention since recent studies have reported the detection of the SARS-CoV-2 RNA in feces, urine, and wastewater. And I will talk about these in a moment. And in addition to that, the concentration of the virus in wastewater has been shown to be proportional to the number of COVID patients in a sewer shed, meaning an area where the sewage is coming from. It's not new, by the way, to use environmental surveillance. It's been around since probably the 1990s to monitor the prevalence and movement, circulation of diseases, viruses in the population. But as a result, it is a rapidly developing scientific discipline for monitoring real-time data on geographical and temporal infection disease trends in the population. It's also very useful, and I will show you why in a few minutes, for detection of asymptomatic and oligosymptomatic cases, which may be missed by clinical surveillance. So you're aware, asymptomatic means you're not showing symptoms. Oligosymptomatic refers to those patients who are showing very few symptoms or very mild symptoms. It involves sampling wastewater from communities, where you have communities that are, of course, sewered, or sewage treatment plants. Like I mentioned, it uses the same PCR method that we use for nasopharyngeal samples, but with some modifications for pre-concentration and cleanup of the viral particles. This is done to increase sensitivity, as there are lower viral loads in wastewater and feces urine than in the nasal and throat swabs. How can this be useful? Well, in wastewater-based epidemiology, the prevalence of the virus or the infections in the community could be estimated by enumerating the virus RNA in the sewage and then performing mass balances on virus shedding and using population and sewage flow rate data. So we can use this information then to inform public health responses to the outbreak. And that is the reason I'm giving this talk, because I'm hoping to gain some or to garner some interest in this particular area of study. All right, so what are some of these findings? And I see my time running very quickly, so I'm going to try to get through these as quickly as possible. To be honest, there have been about 30 reported studies, maybe more by today, on COVID-19 in water and wastewater, both in peer-reviewed articles as well as in preprints. So I'm going to try to summarize some of this data. Much of the information, though, you can find at these websites that I've posted here, including the one at the bottom, which was a publication from the WHO back in July 2020. Most of the data I talk about here are summarized in this article as well. So what are the findings? As I mentioned to you already, SARS-CoV-2 has been found in feces and urine, and definitely in the feces and urine of infected patients. So we've seen positive, or scientists have seen positive results. And I have found more than 10 studies showing positive results for SARS-CoV-2 in the feces and urine of patients in the short time that this has been studied. An interesting finding is the detection of the virus in feces of patients up to 5 weeks after their respiratory samples tested negative. This suggests then that persons are shedding the virus for weeks after they've stopped showing symptoms, or weeks after they're even sick anymore. Mainly fragments of the virus have been found in the feces and urine, but 3 of the studies that I found, that I read, found live infectious virus in feces and in urine. Other studies have shown that the virus can persist up to 3 days in feces and 17 days in urine. And like I said, though, most of the time they've seen fragments of the virus, and that is because the virus does not appear to be resistant or very resistant to the environment of the gastrointestinal tract, unlike the enteroviruses like E. coli. So much of the virus breaks down, or the virus tends to break down as it passes through the GI tract. Here are some issues, though. The mere fact that there is, we've been able to, or persons have been able to detect the virus in feces and urine, and some live virus has been found, it means then that fecal oral, as well as urine oral, and fecal respiratory, as well as urine respiratory transmission is possible. That's not a good sign. And this is especially the case if the urine, feces, or sewage becomes aerosolized. So it would be a good idea to close the toilet lid when flushing. And for persons cleaning bathrooms of COVID patients, those persons need to be extra careful. So what about in wastewaters? Again, SARS-CoV-2, as you can imagine, has been detected in wastewater, and this has been detected in at least 11 countries, and these include China, a number of countries in Europe, Brazil, the US, Australia, Turkey, Japan, and even India. A recent study came out from a colleague of mine in India. Actually, it hasn't even come out yet. It's been published. It was accepted this month. In all of the studies, they observed reductions in the virus RNA load over various stages of wastewater treatment. So what happens is that untreated sewage and primary sludge showed the highest viral loads. And primary sludge actually showed the even higher than sewage itself. And the thinking is that, like bacteria, viruses adsorb to the surfaces of solids, and the sludge is a solid part of the sewage. So you can imagine that the virus would adsorb to the surface of the solids, and therefore the sludge would tend to have higher viral loads. So the studies also found the virus persisting up to 3 days in hospital wastewater and domestic sewage. So like I said, there were decreases in the loads over the wastewater treatment. So lower viral loads were found in secondary treatment. And in that study coming out of India, in some cases, especially when aerobic treatment is involved, the viral loads drop to undetectable after primary treatment. None of the studies found or detected the virus in tertiary treated sewage and disinfected wastewater. And when I talk about disinfection, I'm talking here about chlorination or addition of peracetic acid. So although we cannot say that treatment is 100% effective, there is certainly evidence that supports a significant drop in the viral loads in wastewater following various stages of treatment. So what is happening is that the virus shows poor stability in wastewater and therefore more than likely low transmission potential. And I say that possibly, right? Possibly low transmission, because remember I told you that 3 studies did find live virus. Survival of the virus depends on a number of factors, including temperature, the presence of organic matter and solids, and the types of microorganisms that are present. But some studies showed a 99.9% reduction within 2 days of the viral loads within 2 days in primary sewage. And a very important part of all of these studies that I found, I guess from a researcher point of view, I can't help myself, is that in one study in Brazil, virus DNA was detected in sewage samples that were collected back in November 2019. Now, pay that date in mind, I think it was November 27, 2019. This was 60 days before the first reported case in South America and 90 days before the first reported case in Brazil. So what does this mean? It means then that it is possible to detect the presence of infected persons in the population before people even know they're infected, and certainly before there is an outbreak. Another important finding is that the viral loads in the wastewater increased as the pandemic worsened in these areas. So based on that, we know then, and based on this diagram, you can see that there are pathways for the transmission of the virus from wastewater into the water environment, the aquatic environment. So then I'm sure your next question is, are our natural water bodies safe? Again, studies have been conducted on our natural water bodies for various coronaviruses. I'm not sure about other areas, but there is one study in particular in Italy that found viral loads for SARS-CoV-2 during the height of the outbreak in Italy in some untreated waters and rivers. The viruses that were found may or may not have been linked to discharge from wastewater treatment plants, but certainly that would be a source: untreated sewage, runoff, broken main sewage lines, stuff like that. And to be honest, other coronaviruses have been found in other rivers around the world, so it's not really surprising that the SARS-CoV-2 would be found in rivers where we have the infections in the population. Is there a potential for transmission? Well, we know that the survival of the virus depends on all those factors I mentioned. Time is going quickly. All those factors I mentioned, including temperature, solids, organic matter being present, types of microorganisms. And we do know, though, that the virus can survive for a number of days in wastewater and other types of water. So there is a potential for transmission, but at present, the WHO does not think that there is a great potential for transmission, because most of the time there is only fragments of the virus, and therefore virus that is not viable. Still, we're learning more about it, so we really need to do more testing in water bodies to evaluate whether or not this is a strong potential for transmission, and therefore to develop appropriate risk management strategies. A big question, of course, is whether or not our drinking waters are safe, and I've grouped it with recreational waters. And to be honest, though, thankfully, the SARS-CoV-2 virus has not been detected in domestic treated water in these studies. Now, I haven't seen any reported studies for Jamaica, so I'm telling you about the studies that I've found in other countries. But I would imagine that to be the case in Jamaica as well, because drinking water here and in other countries are typically disinfected with chlorine. And I think you are aware that chlorine is a very effective disinfectant for this and other viruses. The question, though, is what about other sources of drinking water, such as trucked water, bottled water? This is where it gets a little tricky. These may or may not be safe. Basically, all the sanitation practices in these water industries need to be strictly adhered to. And in fact, we need strict sanitation practices. These industries need to pay attention to sanitation in their factories, in the handling of the water from source to public. Water handlers, particularly like truck drivers and those dispensing water from their trucks to tanks, need to wear gloves, masks, all of the similar PPE that you would imagine. Because if anybody is infected, then they could potentially transmit the virus to the water body, and then persons will get in contact with that water body, will get in your hands, etc. And I mean, washing hands is always a good idea, but and that is a good way to prevent the spread of the virus, but certainly this is a concern. Even if the water is disinfected. So even if you disinfect, if you get your tap water and store it in a tank, if you're storing it for an extended period of time, you're going to want to consider disinfecting it by adding a little chlorine bleach, as you would normally do to disinfect water. What about pools and recreational waters? Again, like I said, the WHO thinks there is a low risk of transmission in these types of water bodies. And if the pool is properly maintained and adequately disinfected, it is thought that it should be low possibility of transmission. But properly disinfectant means a minimum free chlorine level of 1 milligram per liter. However, even though water may not be the natural water bodies may not pose a very significant risk for transmission, small crowded areas, such as in changing rooms and showers, and of course, surfaces that may be touched, can still transmit the virus. All right, so in conclusion, what we have found, or what scientists have found so far, is that the presence of SARS-CoV-2 virus in sewage, and it compares well to the prevalence of the disease in the population. The virus found in untreated sewage may or may not be infectious. And in fact, treatment has been shown to be very effective. Maybe not 100% effective, but certainly there is a significant decrease in viral loads after different stages of treatment. Sewage surveillance can be a useful public health tool and could serve for early warning and to anticipate outbreaks, map the circulation of the disease or infections in the population, as well as an increase or decrease in infections. And when I say that we could use this could be used on a local scale, a community scale, or even a regional scale. So local as UE could start doing this sort of testing on the wastewater leaving dorms. So for example, if there's a point at which you can sample, or we can sample the wastewater leaving each dorm, then we can be monitoring the wastewater leaving the dorm and catch if there are any infections in that dorm before persons even start showing signs of the disease. And of course, this can be done for communities as well as on a regional scale. The one limitation, of course, is that we need to be able to collect the wastewater. So it's really applicable to communities that are sewered. What can we do? Certainly we need to protect our drinking and recreational water sources, which means we need to avoid the disposal of fecal matter or sludge or untreated wastewater in and around our water bodies. We should drink treated, disinfected water, and also pay attention to how we store our water, how we handle bottled water and trucked water. Certainly, we know that viral loads in sewage decreases at each stage of treatment, but we should consider adding a disinfection step to be sure. And of course, contain all spills, contain the sludge, and even treat the sludge if possible. In our households, infected persons should use a separate toilet where possible, and definitely we want to fix all faulty plumbing and sewage lines. And there was a study out of China that showed infections in an apartment building that occurred because there were broken wastewater lines in the building. And in other words, the wastewater or the sewage became aerosolized and infected persons throughout the building. So that's a concern for us, persons who live in apartments. Chlorine bleach, of course, is always a very effective way of disinfecting, and of course, boiling. Sanitation services and their workers must carefully handle treated or untreated sewage, so proper PPEs, safety precautions, and they should be doing not only the regular testing that they're doing now for other diseases like the enteroviruses, but also they should consider the testing of their personnel for COVID-19. And what I would like to see happen here, as is happening in other countries, is for us to start environmental surveillance for SARS-CoV-2 in wastewater and sludge, meaning wastewater-based epidemiology, and of course, in our rivers, streams, etc., that may be impacted by untreated sewage. And this is where UE can help. And just to end here, there are some other areas of research that are being explored or suggested. Research into the development of new or to upgrade existing potable water and wastewater treatment facilities to handle this virus and other viruses, and particularly when it comes to wastewater coming from hospitals, clinics, nursing homes. There are specialized virus inactivation treatments that are being investigated or being suggested, which include UV treatment, ozone-based technologies, and photooxidants, of course. We could also look at examining the efficacy of various disinfection agents and the technologies for disinfection, which include some of these same technologies. There is even on a different scale where we could look at household drinking water disinfection devices. You know, just like how you have a filter on your pipe, maybe there is potential for development of disinfecting devices that could be attached to or placed in our homes with UV like a UV point of view system. And this is where we can assist in chemistry. Of course, we will need to establish partnerships with other departments and local government sectors, international partners to do some of this research here. And that's where I will end. And I'll just remind everyone to stay safe, stay home, and save lives. Thank you.
Thank you very much, Dr. Gordon-Smith. Now, from wherever you are, we can give her a round of applause.
Thank you. I'm sorry I went over time there.
That is certainly relevant to all of us in our own lives. And certainly as chemists, you've brought a lot to us in terms of other ways that we can contribute to the science that is evolving to arm us in the fight against COVID-19. So we have 10 minutes or so for some interaction, and I see Prof. Mulder. I think Prof. Mulder has put something in the chat, if I can bring that up here. Dr. Gordon-Smith.
I'm seeing it, yes. Thank you for that, Prof. Mulder. I actually have been meaning to contact Dr.
Dr. G, just read that comment for me, please.
Okay, it says, Prof. Mulder here says, I know that Dr. Joshua Anzinger in the microbiology department has very recently conducted a study on sewage from UE Hospital. Initial analysis didn't detect the virus. Samples were probably too diluted. Yeah, that's one of the issues with wastewater, Prof. And I really need to get in touch with him. There needs to be some pre-concentration and cleanup of the wastewater to increase the viral load so that you can actually detect it. And that's probably what happened in addition to the fact that they only have a certain number of patients who actually have COVID at the hospital, then you can imagine it is diluted down even more. So that is an area in which we can definitely help. And so that study I mentioned out of India that is being published, that was accepted this month, they outlined some of what they did in the analyses to pre-concentrate and clean up the wastewater before PCR analysis. I'm happy.
Do we have any more?
Definitely want to talk to him.
Any more questions? Any more comments, everybody? Anybody? I was interested in this, in the fact that it's in urine and it's been detected and lasts for up to 17 hours in urine. And I thought about
17 days actually.
17 days, right?
Yes.
Sorry. So I thought about the pH effects on macromolecules, pH and protein stability and activity, and you know, just I think acid-base unfolding of these proteins, the spike proteins, and how that affects the interaction with the cell membrane, because I think it's the spike proteins that interact with the cell membrane, and so if we can interfere with that process. Are there any studies on the pH effects on the specific spike proteins that are on COVID-19?
To be honest, I'm not aware if there are any specific studies. There are probably other studies on that with other coronaviruses. But that is something that definitely is an interesting area that could be looked at in terms of disinfecting and as you said, yes, and combating the spread of the disease in that way. I mean, that's how the disinfection agents work. Anyway, it interrupts the ability of the virus to either survive with the lipid cover or interferes with the interaction between the virus and the cell membrane. So yes, I think that is a very useful area. And if we look at it, we'll probably find other studies on coronaviruses in that way.
Right. I see Onero. Onero, you have your hand up. You may open your microphone and go right ahead.
Thanks, Dr. Gardner-Smith. A really informative presentation. My question though is, you indicated that the virus was detected in feces, both fragments of it as well as live viruses. What I would want to know, did they suggest, or is there a link between this stage of the person in terms of them contracting and whether should I put this now?
And yeah, and if the viral load changed, you know,
well, not just that, but in terms of I think some persons have expressed that they have diarrhea initially prior to find out that they have COVID. So I would like to know if there is a link between persons being in the early stage of contracting the virus and them finding live viruses in the feces compared to when persons have probably recovered and their saliva sample would have tested negative. If that's the case, when you will find fragments of the virus in the feces. Did they say anything relating to that?
That's an interesting question. To be honest, on air, I don't recall. I don't think so, but I don't want to say that for sure. I don't recall in the study that I read them relating when they were infected to when the feces was found, up to when the virus was found in the feces. Other than that, it definitely was present when they were contagious and infected, as well as after when they tested negative. I don't quite recall, but I can share the studies with you. Or in fact, if you go to that WHO, I can share it with you so you can read more about it. Because to be honest, I don't quite remember if there was, if they saw that correlation. But certainly, one of the reasons I've read why persons may get diarrhea or other, stomach pain, etc., is that some of those same ACE2 receptors that you find in the lung is found in the gastrointestinal tract. So therefore, you can imagine the virus can also affect the stomach, the intestines, etc. And maybe that is what is leading to some of the other symptoms like diarrhea and stomach pain, etc. But no, to be honest, I can't answer your question. I'm not quite sure if that was the case from that study. And it was only one study, by the way, that I found that information about live virus versus not live virus. No, sorry, three studies.
Okay, but as in, I think that correlation would be interesting.
It certainly would be.
Yeah.
It
All right,
would.
thanks much.
Okay. Do we have any more comments or questions? Is there anybody else who would like to make a contribution? I'm not seeing anything else. Are you seeing anything else, Dr. Gordon-Smith?
No, I'm not. It was either very clear or very boring and everybody's sleeping.
Or people are just digesting this information. And so, all right, so we just have about 4 more minutes. So in that time, I'm going to ask Dr. Maraj, are you there? Could you just help me to welcome the new graduate students who have joined our department before I give the vote of thanks to Dr. Gordon-Smith? There are some new graduate students who are here in the department. There are 3 of them, right, Dr. Maraj? Okay, not hearing from him. So if you're out there, I'm not sure who the 3 are, so we will leave that until next week. Dr. Gordon-Smith, we are going to close the seminar this morning, and it's only left for me to say once again how much I appreciate this talk this morning, how much I appreciate you taking the time to share with us. I know it's a busy time for everybody, and we appreciate the way you have broken down the information, reviewed the literature, and brought to us not only things that are relevant in our daily lives as individuals, but also opportunities that are there for us as chemists to contribute to this fight against COVID-19. I want to thank you again and ask everybody to stay where you are and give her a virtual round of applause. If we were in C2, we would have presented you with a little token.
That's okay.
So you are missing out on that one, but I hope you're feeling the energy and the gratitude, Dr. Gordon-Smith.
You're most welcome.
I want to say before we say goodbye, guys, I want to have you look out for the talks that are coming up. Next week, Dr. Alison Gajada will talk to us from St. Lucia. She is in the audience today, I see, so welcome, Dr. Gajada. Dr. Catherine Morey will speak the week after from the UK. We have Professor Amal, who is coming to us from Egypt. She is the head of Women in Science Without Borders. We have Dr. Kenisha Wilson, who will be coming to us from the United Arab Emirates. So we have a number of very interesting talks that are coming up. I wish you a safe and productive week and ask you to join us again right here next week, same time. Goodbye, everybody.
Bye.